Thank you for listening to my poster presentation, where I'll be discussing my research regarding how perinatal iron deficiency causes sex-dependent alterations in kidney retinoic acid signaling during development. To provide some background, the developmental origins of health and disease hypothesis posits that stressors during development can impact lifelong health and organ function. The stressor our lab studies is iron deficiency, the most common nutritional deficiency worldwide, affecting over 2 billion people, with pregnant women most at risk. Iron deficiency has been associated with impaired kidney development, specifically reduced nephron endowment, but the mechanisms are not well known. Nephrons are the functional unit of the kidney, and the number of nephrons endowed during development stays the same throughout the lifetime. This can have implications on long-term cardiovascular and renal health. Retinoic acid is a metabolite of vitamin A and an important determinant of nephron endowment. That brings me to my hypothesis, that perinatal iron deficiency causes a reduction in nephron endowment by disrupting retinoic acid signaling. We used the rat model of iron deficiency where 2 weeks prior to mating, female Sprague Dawley rats were either on an iron-restricted or a controlled diet. Offspring kidneys were collected on postnatal day 1 and 28, and they were flash frozen. To quantify the number of nephrons, we used a technique called kidney acid maceration, where the kidneys are extracted and digested in hydrochloric acid, and the glomeruli are left behind to be quantified. To look at the expression of some genes that encode elements in the vitamin A pathways that dictate nephrogenesis, we used quantitative polymerase chain reaction. Moving on to the first set of graphs, we found that maternal iron restriction caused a reduction in neonatal body weight at postnatal day 1 and 28 in both males and females. This confirms our model indeed impairs growth. Additionally, we found maternal iron restriction to cause a reduction in neonatal hemoglobin at postnatal day 1 in males and females, but this was replenished at postnatal day 28 due to the standard diet the pups are put on after birth. To our main results, iron deficiency caused a reduction in nephron endowment at postnatal day 28 when all the nephrons are done being formed. This only was observed in males. Next to the graph is a representative image of how the glomeruli look after acid maceration, as can be seen by the red arrows. My next figure gives an overview of retinoic acid signaling. Retinaldehyde is converted to retinoic acid by the enzyme retinaldehyde dehydrogenase 2. CYP26B1 is an enzyme that inactivates excess retinoic acid, but retinoic acid mainly exits and gets transported out of the interstitial cells to activate receptors on the developing ureteric bud on the kidney. The downstream effector CRET ultimately leads to kidney growth. On the top right, we measure gene expression of some of the main signaling components at postnatal day 1 and 28. At postnatal day 1 in the kidney, in the interest of space and time, I'll only be presenting some of the most notable ones. We found that iron deficiency caused an increase in retinaldehyde dehydrogenase 2 expression, CYP26B1 expression, and retinoic acid receptor alpha expression, predominantly in males. It didn't affect the expression of downstream effector CRET. At postnatal day 28, iron deficiency similarly increased expression of retinaldehyde dehydrogenase 2. However, what was interesting was that CYP26B1 and retinoid X receptor alpha were upregulated in females and not due to iron deficiency. We found that iron deficiency caused an increase in the downstream effector CRET at postnatal day 28. So to summarize, at postnatal day 1, iron deficiency caused increased expression of many components within the vitamin A signaling pathway. These enzymes are important in determining nephron endowment, so this overexpression may indicate an attempt to compensate to perturbations caused by iron deficiency in order to maintain signaling. At postnatal day 28, we see similar trends with retinaldehyde dehydrogenase 2 and CRET being upregulated. Notably, at postnatal day 28, females also had many genes that were upregulated, including retinoid X receptor alpha and CYP26 enzymes. This may indicate sex differences in overall retinoic acid metabolism, or it could be that these enzymes play protective roles in the cell and thereby explain no reduction in nephron endowment in the females. Just wanted to say thanks to all of my sponsors and the people in my lab that helped.